All right. Well, it's a pleasure to have Linden Parkes with us this morning. One of my former PhD students, an all-round great guy, and he's been doing some all-round great science, especially since he stopped working with me. And one example of that is this paper that I saw and read through and thought it was great work and invited him to come and present to us. And so here he is presenting the paper on your screen. So yeah, thanks very much for making the time to present to us, Linden. Looking forward to your talk.
Cool, thank you. Well, thank you very much for the invitation. It's really good to be here. Can you guys, cool. So I think a few of you have seen this talk in a couple of forms or talks like it. So I think, for example, I came and gave a sort of rough version of this to Mac Shine's group a little while ago. So yeah, it'd be great to see if some more thoughts come up on a second viewing in a different form, and I'm looking forward to any questions people might have. But today I'm going to, so as Ben mentioned, I'm going to be talking about this paper that we just published in Science Advances late last year called Asymmetric Signaling Across the Hierarchy of Cytoarchitecture within the Human Connectome. But I'm also going to kind of tie in a few other bits and pieces of my work that are kind of relevant to what I think about and sort of inform the reason why I did this project and where I kind of drive my work towards. What's going on here? All right, so a big central goal of my research has been kind of really focused on, improving our understanding of how we kind of map the signs and symptoms of mental health to the brain. And while the paper we're here to discuss today doesn't focus on this problem directly, I like providing this context because it sort of shapes the questions I've been asking in my work, which is relevant to this paper. And also today, as I said, I'm going to touch on some findings from my previous work, as well as some preliminary data going forward that kind of link in both sides of this work, of this paper here today, that kind of speak more directly to that goal of biomarker mapping. And in studying this mapping between psychopathology and the brain, I, for the most part, adopt a connectome approach, wherein all the connections between all the regions in the brain are represented as a network like this. A sort of very quick primer for those unfamiliar with these network neuroscience terms. In the human connectome, we use kind of two basic key terms to describe its components. First, we think of the brain regions as nodes of that network, which is the orange circles here. And these are defined typically according to some parcellation of the brain, which of course there are tons that you can choose from. And second, we think of the connections between these nodes as edges of our network. And it's important to note that edges in particular can be defined in several different ways, and how we define those edges informs and constrains the types of questions we can ask while studying the human connectome. But for all the work I'm going to present today, we're going to consider edges defined according to tractography performed on diffusion imaging. And so defining edges in this way gives us an estimate of the number of physical white matter pathways that connect two regions. And so today we'll be talking about weighted structural connectomes and everything. And casting the brain as a network in this way kind of really opens up a lot of great opportunities to study the brain through its topology, or the topology of this ensuing graph. We can ask questions about how the brain is connected, what regions connect to different regions different ways, how those groups of regions sort of group together into communities, what regions may be more or less important in the brain, sort of how, and ask questions about how the brain maybe integrates information. And we can ask questions about how the kind of complex pattern of connectivity enables things like dynamics to unfold on the network. And all of these things I'm sort of interested in leveraging these tools to kind of map changes in individuals who experience mental health. So, the big question for me is like, how does connectome topology map to psychopathology? And the field has been probing this question for some time now. I'm by no means the first to do it. And there are lots of different subdomains of network neuroscience that address this question from a bunch of different angles. And what I'm going to focus on today is kind of two approaches throughout my postdoc that I've been working on in a kind of reciprocal fashion of sorts to try and probe this general idea. So the first is a subfield of network science called network control theory. In essence, network control theory involves taking the structural connectome that I mentioned and sort of layering on top simulated dynamics at every node. So I'm illustrating the simulated dynamics using the orange time series here. And then control theory then involves asking questions about how the topology of the system supports and constrains the spreading of those dynamics along its connections. Also, as the name implies, control theory is also concerned with modeling control signals, which I'm illustrating here with the blue time series instead. And what we do is we inject those control systems into this now dynamical system, and then we in turn see if we can use those control signals to effectively sort of steer or control the spreading dynamics and get them to go where we want them to go on the system. So the orange lines represent our sort of simulated dynamics that if they're uncontrolled, they'll just spread throughout the system in a diffusive manner and would, constrained by underlying white matter pathways. And the blue time series are an additional set of time varying signals that we model and we inject into the system to try and control the dynamics. And the second idea is this emergent idea of a biologically annotated connectome. This is sort of growing in popularity, and today the annotations I'm going to be talking about are going to be based upon Cytoarchitecture, which is an index of regions, sort of layer-wise cellular composition. Now, so one of the great strengths of network science or the network science approach is that it creates a nice common language to understand complex data across multiple scales and domains. Today, we're talking about macroscopic brain networks, but we could specify these connectome graphs using brain data at a very different scale or spatial scale, for example, or measured in a different animal or in a totally different data type, like a social network. And in all of those cases, once we have a graph of nodes and edges, we have a nice common language for describing similarities and differences between those systems. However, the cost of that power, if you like, is that the graph representation abstracts away a lot of biophysical detail of the brain. And often in the analyses of brain graphs, nodes are sort of treated as though they're all the same. And so the idea of a biologically annotated connectome addresses that issue by trying to add back information about regions' biological properties and asking how imbuing the network with that knowledge helps us to better understand topological properties. And it's sort of here at this sort of lower half of this picture that this paper sits, which is the idea of trying to integrate network control theory with a biologically annotated connectome to study individual differences. So what I'm going to do now is walk you through some of the main findings of this paper, and also, as I said, some of the related work about predicting psychopathology. But first, just a quick primer on the network control theory model. It's quite a simple model that follows this equation here. I'm going to try and unpack it a bit to build some intuition. So the model is a very simple linear dynamical model where the output simply tells us how the activity at each brain region changes over time, which is stored in X dot. And this change in activity is a function of two factors. The first is the internal brain dynamics. We touched on this before, where activity diffuses from one region to another along the structural tracks. And then the second factor is that additional control input that we provide, which you can think of as either a sort of simulation of external stimulation protocols or like a internal cognitive control, if you like. So the model simply simulates the evolution of brain states as a function of both internal diffusion as well as external modeled control that we seek and we find from the model. So one of the common things that we can do with this, and this has been done in the past, is we can basically define pairs of brain states that are of interest to us as neuroscientists, and then we can try and find those control inputs that move activity from one brain state to another. And then we can study how the topology of the network made that transition possible. For example, this is a figure taken from Rick Betzel's work from a few years ago. We might be interested in the types of control signals that we need to model in order to move the brain from, say, an initial state where the somatomotor network is active to a visual state where the visual regions are active. But importantly, this transition from somatomotor to visual may not be sort of natural for the system, given its underlying structural connectivity and dynamics. And so in order to achieve this, what we want to do is find the control signals that will move the system from one point to another with the least amount of effort or the lowest amplitude. And if we ran, the model without those control signals, then we might end up somewhere else, where, say, for example, here in a different pattern of activity at the end of the simulation. So once we've modeled some control signals, we use them to estimate what we refer to as control energy, which is the integration over time and the summation over nodes of the control signals we find. And this just kind of gives us an estimate of the amount of effort the model had to go to in order to make the transition occur. Importantly, this metric is not necessarily symmetric. That is, the energy required to go from one state to another may not be the same as going from the target state back to the initial one. And this is because the control signals can leverage kind of different aspects of network topology when trying to drive dynamics in one direction compared to another. And that's something we're going to see today in the Science Advances paper. Okay, so we have our model. How do we annotate it with biology? So there's a really long history of evidence from neuroanatomy that tells us that brain regions profiles of cytoarchitecture informs how pairs of regions are structurally connected. So as I touched on earlier, briefly, cytoarchitecture is the study of the cellular composition of brain tissue. So in the cortex, it involves studying the makeup of different layers within the cortical surface, which is pictured on the left here for 5 different regions. And broadly speaking, it involves examining how many layers different regions have, how clearly defined they are, and what the density of different cell types is within each of these layers. And it's broadly thought that different regions have different cytoarchitectonic types, and that this plays a role in their connectivity and their function. And so this research tells us that the kind of interregional similarity in cytoarchitecture plays a role in determining the pattern of feed forward and feedback projections that connect pairs of regions, and it makes predictions about asymmetries in these projections between region pairs. So in short, cytoarchitecture seems to tell us something about the nature of structural connectivity. And a very sort of exciting recent advance for me has been the release of the ultra high resolution histological atlas called the Big Brain data set, which is pictured here on the top right. This atlas has recently been summarized as a gradient, so another gradient in the great space of neuroimaging gradients, which is shown down here in the bottom right-hand corner. And this gradient, this is work from Casey Paccola in some of her excellent papers. This gradient basically describes the extent to which pairs of regions have similar or dissimilar underlying cytoarchitecture profiles. And so this map allows us as network neuroscientists to integrate our network model with a precise map of how regions profiles of cytoarchitecture vary across the cortex. And this opens up lots of cool opportunities to link topology of the connectome with these microscopic features that we think control or govern extrinsic connectivity between them. So in this paper that we're talking about, basically what we wanted to do is we wanted to use control theory to ask whether and how cytoarchitecture constrains topology of the macroscopic connectome, and whether and how that informs brain dynamics in our linear dynamical model. Specifically, we reasoned that if cytoarchitecture explains asymmetry in extrinsic structural connectivity at the microscale, it may shape patterns of macroscopic connectivity and shapes the topology we see in these networks. And in turn, we might expect topology of the network to result in asymmetries in the way dynamics from our model traverse the connectome, and that these asymmetries may be explained by differences or variations in underlying cytoarchitecture. And then in turn, if those asymmetries are biologically meaningful, we also reason that they should show developmental effects in a developmental sample we have, and I'll get back to that and explain that in a moment. So to examine this kind of broad question, we started by sampling groups of regions that have similar profiles of cytoarchitecture. So this is taken from figure 1 in the paper. This gradient on the left is that gradient of cytoarchitecture from Casey Piccola's work. It's the same one from before. I'm just showing it with a different color map here. So basically, if I say sample 2 groups of regions that are at different positions on this hierarchy, as I'm showing here, then both small groups will have, relatively internally similar profiles of cytoarchitecture because they're close together on the gradient, but those profiles will be different between the 2 groups. And importantly, they become more different as the distance separating them along the hierarchy grows greater, right? So if we sample 2 groups, one group at either end, they'll be very different. We, say sample a pair of groups nearer to each other, they'll be relatively more similar. And so what we do in the paper is we sample sets of regions from this hierarchy, and we take those small groups and we carry them forward into our control theory model. And we treat them as kind of brain states in air quotes, right? They're not states in the sense that they're like patterns of activity, but what it allows us to do is it allows us to ask, well, if activity was seeded in one set of cytoarchitecture or one set of regions with a certain profile of cytoarchitecture, and they had to communicate with a different set, how would they do that? And so this basically lets us examine transitions or state transitions in control theory language between patches of cortex that are located at different positions along this hierarchy. And importantly here, what we're arguing is that we can model bottom-up and top-down state transitions based on the idea that one patch of cortex is always lower than the other on this hierarchy or vice versa. And so if we have lots of different patches of cortex along this hierarchy, we can estimate the transition energy between them all in both directions, and we can easily group those transitions into bottom-up and top-down. And then by subtracting bottom-up and top-down, we can get an index of how asymmetric that transition energy is. Because remember before I said this is not necessarily a symmetric measure. And this allows us to basically study, whether it's easier or harder for activity to be propagated in one direction or the other across this version of the cortical hierarchy. So when we do this, we find that in general, bottom-up state transitions are easier to complete. So this is from figure 2 in the paper, or it's sort of an abridged version of it. So this plot on the left shows results that we get when we run the model on just a group average connectome taken from about 700 subjects. And it demonstrates that the energy associated with moving bottom up the hierarchy is significantly lower than the top down equivalents. So this suggests that sort of propagating dynamics up the hierarchy is easier for our model compared to down. More importantly, when we actually subtract these pairwise energy estimates and look at the differences, we find that the distance separating the brain states across the cytoarchitecture gradient correlates negatively with the energy asymmetries, which is this plot in the bottom left hand here. So here in this plot, every point is a specific transition that we model, and the y-axis is the energy asymmetry, and a negative value, which most of these are, indicates that bottom up is lower than top down. And then the x-axis is the distance separating states on the cytoarchitecture hierarchy. So this negative correlation tells us that the more dissimilar cytoarchitecture is between pairs of states, the more negative those energy asymmetries become, or the easier it becomes for the bottom-up version to complete compared to the top-down. These plots here on the right and both the top and bottom panels are a pair of null networks that basically demonstrate that these effects are not better accounted for by combinations of spatial embedding and brain regions, the edge weights of the network, or brain region strengths. So this is just based upon, you take the network, we reshuffle the edges, say 10,000 times, subject to a set of constraints based upon space and other properties. And we find that the effects, both the asymmetry itself and this negative correlation with space, are not simply accounted for by the things we preserve in our null. So we think this is a nice finding because it kind of fits with the idea that regions pattern of feed forward and feedback projections, which is shaped by similarity and dissimilarity in cytoarchitecture, that this governance over extrinsic connectivity may be reflected in the macroscopic level in network topology. And we can sort of probe that with network science tools. So the next question that's obvious is, how is this happening? What are the features of topology in the graph that are enabling this to happen? So to address this question, so this is now from figure 3 in the paper, we drew from sort of several other metrics of network science that probe how regions communicate with one another in an asymmetric way, and we correlated them with our energy asymmetries. So we'll start slightly out of order with this figure. If we look just at the bottom left 2 for now, basically what we found is that energy asymmetries correlated negatively with asymmetries in diffusion efficiency. And diffusion efficiency is just a measure of, if you have a diffusive model like we do, and you start at one node and go to the other, how efficient does that diffusive activity spread to node J? And we also find a positive correlation with a metric called search information, which measures the likelihood that a random walker starting at node I will reach node J via the shortest path between node I and J, right? And so if a random walker is very likely to hit node J from node I via the shortest path, then the search information is low. And so what the take home really here is that basically these results show us that state transitions with lower bottom up energy relative to top down are also associated with more efficient bottom up diffusive signaling. And that is maybe in part because they're more likely to find the shortest paths when activity moves up versus down. That's what the search information tells us. We also studied a variant of path transitivity, which is not typically asymmetric, but we modified it to create an asymmetry. So path transitivity is such that basically, if you take a shortest path between a pair of nodes, like I'm illustrating here in panel A, path transitivity measures the extent to which activity activity deviates from, but then returns to the shortest path. And so this is measured as like the presence or absence of returning detours of a certain length. So as an example here, these, if you're moving from S and you're moving through A to B, and these paths branch off, but they don't return back to the shortest path, versus up here, there are these little triangles on the path where activity might come off the path and then return later. So path transitivity measures the extent to which these sort of returning detours occur along the shortest path. So what we did to introduce an asymmetry, because this is typically a symmetric measure, is we examined the cumulative transitivity of a given shortest path, starting from each end, right? So basically, so what we did is we calculate path transitivity for bigger and bigger chunks of a shortest path, starting from node S, but also starting from node T, so going in both directions. And this gives us basically an estimate, so we end up with a variable where the path transitivity accumulates as we get larger and larger chunks, right? But they can accumulate at different rates depending on where those little returning detours are across the path, right? So in this example, the returning detours are closer to T, closer to node T than they are to node S. So they're getting counted sooner. And so the accumulation of path transitivity is higher from running from sort of T to S versus S to T. So basically we end up with these two curves of path transitivity, which we subtract and then sum the differences. And then that gives us an estimate of the extent to which path transitivity accumulated more quickly going in one direction or another. And what we find when we correlate that measure with our energy we get a positive correlation as well. And this, so basically this tells us that these returning detours are kind of encountered sooner for the top-down signaling, which indicates that, so lower bottom-up energy is basically associated with lower bottom-up cumulative path transitivity, or higher top-down. And this tells us that these returning detours sort of higher up on the hierarchy may be inhibiting efficient top-down signaling, if you like. The activity is more likely to sustain and dwell at the top of the hierarchy. Okay, so we have some asymmetries in energy that we can interpret topologically. Earlier on, I said that if these energy asymmetries are biologically meaningful, then they should also show developmental effects. Why did I say that? So the main reason for this is that the developmental literature tells us that this hierarchical coupling between cytoarchitecture and connectivity is thought to be a product of sort of a staged program of neurodevelopment. And one of the early phases of this process that is happening during prenatal development is neurogenesis, which is the process of cells migrating up to the cortex. And neurogenesis is thought to sort of lay the foundation for this variation, this hierarchy of cytoarchitecture. Following that, synaptogenesis then uses cytoarchitecture as a blueprint to kind of guide the refinement of connectivity between regions. And during synaptogenesis, lower order regions sort of refine their connectivity earlier than higher order regions. And so there appears to be a sort of staging of connectivity refinement moving up this hierarchy. And finally, neuroimaging research tells us that this kind of hierarchically staged pattern of connectivity refinement continues well into youth and well beyond birth, and well into the age range of our sample, which is kind of 8 to 22 years. So given this, we expected that our energy asymmetries should vary systematically as a function of age in our sample, because this process of connectivity refinement is still actively happening. And this figure, so I'm skipping now straight to figure 7 from the paper. This figure shows we found exactly that. Here, what we've done is we've estimated transition energy between our cytoarchitecture states separately in each of our subjects, and then we've examined how they vary as a function of age. So this figure shows quite clearly that age tends to correlate negatively with both bottom-up and top-down energy. But what we find when we subtract the energies and look at asymmetries at a subject level, we find this positive correlation here. And what this tells us is that not only do our energy asymmetries change throughout development, but they actually shrink towards 0 as a function of age in our sample, right? Because they're negative to begin with. So positive, as people are getting older, the magnitude of this asymmetry is getting lower. So, before I was showing that bottom-up energy was lower than top-down, now this effect suggests that development might actually be closing that effect. Now this plot on the right is the age effect of energy asymmetry, as a function of the energy asymmetry we get from the group average connectome. So this just basically shows us that transitions with the strongest energy asymmetries at the sort of group average level also coincide with the strongest age effects. So this illustrates to us that ongoing refinement in connectivity, which we think is blueprinted and guided by underlying cytoarchitecture, shapes the energy associated with our transitions. Okay, so just to quickly summarize some of this. So basically what I've shown you so far is the topology of the human connectome is sort of wired to engender asymmetries in the way signals traverse the cortical hierarchy. And these asymmetries result in bottom-up transitions being easier to complete compared to their top-down counterparts. And they also refine throughout development, which here for us is sort of children through adolescence. There's a bunch of other stuff in the paper. We sort of also show some linkage to regional functional data, like decay and time series, for example, and some other bits and pieces. But those are the kind of main take-home points, I think, for the paper. So far, I've shown you that we can use this control theory model to basically understand a biologically annotated connectome and give us insights into hierarchical signaling. But, you know, I sort of said at the start that I care about psychopathology. So the next question is any of this useful for psychopathology? We actually started probing this question in previous work. So this is from a previous paper I'm showing now, where we took a measure from control theory called average controllability, which you can think of as measuring a region's kind of like general importance to controlling dynamics in the model, irrespective of like a specific state transition. So it's just, you know, how important is a region to governing dynamics? And we use this sort of non-specific measure and alongside a handful of other metrics to predict psychosis symptoms in our developing sample. Simply for time, I'm going to skip over explaining some of these other metrics, but they're listed below if you're curious. But the main take-home message here is that there are, you know, lots of canonical measures of topology that we can extract from the connectome that don't depend on this control model. But this measure of average controllability is the best predictor of psychosis symptoms amongst these competing graph theory alternatives. And indeed, it was also the only one that exceeded a permutation test, suggesting it was the only average controllability was the only predictor that was able to exceed chance levels, which is what we show here. So we published this a couple of years ago now in Biological Psychiatry. And the other thing we did in this paper is we also repeated this prediction model using subsets of brain regions sampled across the hierarchy of functional connectivity, which is pictured here on the left. This is the I guess now infamous gradient from Daniel Margulies, published about 5 years ago. And when doing this, we find that prediction gets better for average controllability as we move up this gradient, which is what the middle plot shows, but we don't see the same effect for regional strength, which is just the sum of edges connecting to a specific region. And so what this tells us is that NCT, or network control theory, this measure average controllability at least, is kind of sensitive to the spatially patterned prediction, at least in the context of psychosis symptoms, which is something that isn't true for simpler metrics like strength. So what about our cytoarchitecture approach? The next thing I was interested in is, you know, whether or not we can use the cytoarchitecture approach I was just talking about to improve this predictive performance even further. So going back to the Science Advances paper, I just want to briefly draw your attention to the idea of bystander regions, which are simply the regions that are not a part of the initial or the target state for a given transition. So for, you know, in the case of the cytoarchitecture transitions, this is most of the brain, right? Because we're only really ever transitioning between relatively small sets of regions. One of the details I glossed over about the model is that we implement what's called a full control set. This simply means that all of the nodes of the system, all of the brain regions, are allowed to kind of participate equally in completing a given state transition, irrespective of where they are in the brain or whether they're in and outside of, whether they're a bystander or whether they're in the state or whatever. They're all allowed the same degree of control over the state transitions. So we know that, the bystander regions, which is the bulk of the brain, kind of contribute to the control of state transitions because we let them, but we don't really know how. And this gives us an opportunity to sort of test whether their position along the hierarchy is important to completing these cytoarchitecture state transitions. So what we did here was develop just a simple approach for optimizing the control weights of these control nodes associated with these bystander regions. So this is from figure 6. Briefly, this method basically amounted to giving each region an additional constant arbitrary amount of control and then measuring the change in control energy for a given pair of cytoarchitecture patches. And so basically what we're saying is that regions that exhibit like a larger change or a larger reduction in energy when we give them a bit of extra control, we rank those as more important for a given state transition. And then what we do is once we've done that for every single bystander region, we use that kind of rank ordering to reweight the bystander regions to give them different amounts of control for every, in a kind of bespoke way for every transition. And when we do this, we find that these optimized control weights for the bystander regions correlated with the sensory-fugal axis of cytoarchitecture, the gradient we're using, showing that we're sort of able, this data-driven optimization was able to sort of extract a spatially patterned set of control weights that track the cortical hierarchy. We also found that the size of the energy asymmetries between bottom up and top down became significantly larger compared to our original results, which is what subplot C shows. And so what this tells us is that imbuing our model with knowledge of the hierarchical position of these bystander nodes leads to a significant increase in the size of the energy asymmetry. We think this result is nice because it tells us that, you know, where these bystander regions sit along the hierarchy tells us something about how important they are for facilitating state transitions across the hierarchy. And that if we give the model this information, it actually improves the efficiency of bottom-up signaling even more relative to top-down. But I think the really cool thing for me, and this is what I'm working on presently, is that we also find that this approach improves prediction. So here we've come full circle back to predicting the psychosis symptoms again. And here I've estimated basically both standard uniform weighted transition energy and energy optimized with the method I mentioned at the subject level. And then we've used those two sets of energy asymmetries to retrain a regression model like the one we used for the average control measure to predict psychosis. And so prediction performance in the orange color is for these optimized weights and the blue one is for the standard model. I'm showing a couple of performance metrics here, but they both tell the same story, which is that when we optimize the transition energies, we get a bit better prediction for psychosis, which for someone who spends all day operating in very small effect sizes, mapping the brain to psychopathology, I was very pleased to find. This result's actually not published yet, so this is not from the Science Advances paper, but this is something we're kind of presently working on. Okay, so to sum up quickly, so I've shown you today that network control theory is sensitive to asymmetric signaling across the cortical hierarchy of cytoarchitecture. I've also shown you that NCT seems to improve the prediction of psychosis spectrum symptoms beyond kind of canonical measures of topology, and that integrating this cytoarchitecture approach, or this biologically annotated connectome approach into our model actually seems to improve that prediction a little bit even further compared to our previous work. I just want to close by giving a small shout out to some resources that we've been working on as well. So myself, together with a couple of the co-authors from the paper, Jason and Jenny, have been working on a Python package for these control theory tools, which is currently online at this link. And we're sort of in the middle of writing up a protocols paper as well. So the package is out there, you can start using it, playing with it, giving us feedback if you like, and it'll allow you, if you're a Python programmer at least, to do the types of analyses, all of the types of analyses I've shown you here today, assuming you can bring a structural connectome to it. And the protocol paper will hopefully be able to walk you through a lot of the nitty-gritty details about how to do this in a practical way. But with that, I'll finish up by thanking all of my mentors, collaborators, colleagues, etc., who've contributed in various ways during my postdoc. Of course, thanks to funding services, the NIH and the BBRF, and happy to take some questions.